students, we had started with Phylum Porifera. We had discussed the general characteristics of Porifera. What are poriferans? They are aquatic animals, mostly marine, sessile animals, diploblastic with two germ layers, that is endoderm and the ectoderm. They are characterized by a porous body wall, a central cavity not equal to coelom, and an opening on top. The pores on the body enable uptake of water. What are they called? They are called ostia, which enter into a cavity not equivalent to a coelom because it is not formed by the body wall. The cavity is called the spongocoel. Water enters into the spongocoel through the ostia, wherein the food will be taken up from the water, gases will be exchanged, etc., and the water and the remaining water will be expelled out through the opening on top, which is the osculum. We had also discussed the body wall in detail. The lining around the spongocoel comprises of the choanoderm. The choanoderm is made up of cells called the choanocytes, and the origin of this choanoderm is the endoderm. The choanocytes help to trap the food particles. The outer side of the body wall comprises of the pinacoderm. Origin is the ectoderm. The pinacoderm comprises of flattened cells called the pinacocytes, and they help in protection. The middle layer, an undifferentiated jelly-like substance, is present between the pinacoderm and the choanoderm, that is the mesohyl layer. They do not have the differentiated mesoderm, and the mesohyl layer comprises of the amoebocytes, which are totipotent cells, and they aid in transportation of nutrients, gases, etc. The mesohyl layer also carry spicules and spongin fibers, which comprise the endoskeleton of the sponges. Right? These are the things that we had learned last time. We had discussed the body wall in detail. In this lecture, we are going to look at the water canal system. Phylum Porifera, the water canal system. The sponges are mainly known for their water canal system. It is the network of canals and different passages, pathways which enable the circulation of water into the body of the sponge. The circulation of water enables the sponge to gather food, remove its waste via excretion, gas exchange, that is respiration, and overall circulation. We have studied a very simple passage, that is movement of water through the ostia into the spongocoel into the osculum. This is the simplest water canal system, but there are more complex canal systems in other sponges as well. We can discuss the water canal system as a network of canals and chambers that enable circulation of water inside body of sponge. Look at this is the longitudinal section. As you can see, these are the ostia, and the cavity inside is the spongocoel, and this opening on top is the osculum, a single opening. Ostia are many, osculum is just one. So what happens in the water canal system is that the water enters through these ostia into the spongocoel, and from the spongocoel then the water is expelled out. So the pathway of water in this case is through ostia, spongocoel, and then into the osculum. This is the simplest water canal system that is found in some sponges. There are different types of water canal systems, this being the simplest one. So what is the purpose of this type of entry of water into the spongocoel and then exit through the osculum? So you know that the choanocytes are lined throughout over here, isn't it? Throughout the spongocoel, the choanocytes are lined. So what happens is, with the entry of water, the choanocytes are able to trap food particles, okay, that the water brings along with it. These choanocytes are filter feeders. They will filter out food particles within their microvilli projections, and they will phagocytose them. The flagella that they have will help in creating a water current. The water that is then left over here will be free of the food particles, and then it will be expelled out. Serves the purpose of food gathering, then circulation, gas exchange or respiration. It also serves the purpose of elimination of wastes gathered into these cells. It will serve the purpose of excretion as well. Choanocytes, which line the spongocoel, create water current through the flagella and trap the food particles. They are filter feeders, create water currents using the flagella and trap food particles, okay, using their microvilli processes, the collar of microvilli processes, and that's why they are called as filter feeders. What will they do with the trapped food particles? Trapped food is then phagocytosed. They will do phagocytosis of the trapped food particles inside them, partially digest the food particles and pass the remaining to the amoebocytes that are there behind in the mesohyl layer. Phagocytosed inside choanocytes. So what will you say? The digestion of food, is it intracellular or is it extracellular? It is intracellular. It is internal digestion. Okay, food digestion is internal. Food digestion is internal or intracellular. Interesting to note that the animals before the poriferans, that is the protozoans, okay, the primitive animals, the protozoans also had an intracellular digestion of food through phagocytosis. Even these are known to do phagocytosis of food and intracellular digestion of food. We do not do intracellular digestion of food. We do not take the food inside our cells and then do digestion. The digestion of food is outside into the gut. Okay, the cells will release the enzymes outside and digest food outside into the gut, and the digested food material is then absorbed. But they will absorb whole food through phagocytosis and then digest it inside, intracellular over here. Ours is extracellular. Right about respiration. Their respiration is aerobic, gas exchange by diffusion. Circulation occurs through the water excretion. The water canal system also helps them to excrete out the metabolic wastes that are produced by the cells, and the metabolic waste that is generally produced by the sponge is ammonia. That's why they are called as ammonotelic animals. Okay, we produce urea and we are called as ureotelic animals. They produce ammonia. Okay, urea, by the way, has two ammonia groups combined together to produce urea. We are ureotelic animals and these are ammonotelic animals. Most of the aquatic forms produce ammonia as their metabolic waste. They are ammonotelic, produce ammonia as metabolic waste. Let's talk about reproduction. Reproduction in sponges can be both asexual as well as sexual reproduction via production of gametes. In asexual reproduction, they can develop by the process of fragmentation. A small piece of the sponge, if it breaks by accident and falls off elsewhere, can regenerate into a new sponge. Okay, another method is budding, and another very known method is gemmules. Gemmules formation. Gemmules formation is especially seen in case of freshwater forms, and I would like to talk about the gemmules a bit. Seen in freshwater forms like spongilla. Gemmules are also called as internal buds. Okay, what happens in gemmules is that you know about the amoebocytes that are there in the mesohyl layer, isn't it? What they will do is they will make a compact mass of amoebocytes which will be surrounded by, can you tell me what are these? These are the spicules. And these are the amoebocytes. Okay, and small rounded bodies they will look like, and they will be dispersed into the water. Internal buds they are called as. They will be dispersed into the water. These are known to be resistant to adverse conditions. How do you think these will help in the asexual reproduction? Amoebocytes and the spicules. The spicules will help to protect the amoebocytes that are there inside. They will be dispersed out into the water, and whenever favorable conditions return, the amoebocytes will emerge out and they will recreate a new body of the sponge in that place. Fine, so that is the function of gemmule asexual reproduction. Resistant. Amoebocytes surrounded by spicules. That is what it is. Important. It is there in your syllabus. The difference between gemmules and budding is that budding is like external budding, external buds of the body wall, and gemmules are internal buds. Next we talk about sexual reproduction. Sponges are known to be hermaphrodites. Hermaphrodites means what? Monoecious. Both the sexes present on the same animal body. Okay, they can produce both male and female gametes. However, they prefer to do cross fertilization. Some of the choanocytes will develop into sperms and they will be liberated. The sperms are going to enter into the body of nearby sponges. They will fertilize the eggs of the other sponge into which they have entered. Okay, the eggs, by the way, are present into the mesohyl layer. The fertilization is internal because the egg cells of the sponges are located in the mesohyl layer. The sperms have to travel up till the mesohyl layer, okay, for fertilization to take place. Male and female gametes produced by the same animal body, however, prefer cross fertilization. Sperms leave one sponge and fertilize eggs of another sponge. Fertilization is internal. Why internal? Because since egg is located in mesohyl, Next, development. The development is indirect. The zygote that is formed does not grow into a small sponge. No, it does not. First, it will form a larva, which is very different looking than the adult sponge, and then the larva is going to be liberated from the sponge and it will travel, find a suitable substratum, settle over there, and then it will develop into a sponge. This kind of development is said to be indirect development. Our development is a direct development. For frogs it is an indirect development, and so also for them. Indirect. A flagellated larval stage present. This larva is morphologically means structurally different from adult form. Examples of sponges include Cygon, also called as Scypha, Euspongia, also called as Spongia, which is commonly called as the bath sponge. Then there is one Spongilla, which is a freshwater sponge. Very important examples: Cygon, Euspongia, Spongilla, important examples. There is also one more called Euplectella, commonly called as the Venus flower basket, a very beautiful looking sponge. It is given as a wedding gift to married couples in Japanese culture. These 4 examples you must know for sponges. So with that, we have completed with Phylum Porifera, the syllabus for NEET. In the next class, we will be talking about the different types of water canal systems and classification of sponges. We will also talk about the 2 types of larvae that is part of the AIM syllabus.
महादेव के नाम पे सब्सक्राइब कर दें।